Welcome to Characterization of Therapeutic Proteins for Biological Drug Development. I'm Tamlin Oliver, Managing Editor of Genetic Engineering and Biotechnology News, and I'll be the moderator for this webinar. Biological drugs, including monoclonal antibodies, fusion proteins, and peptides have emerged as promising therapeutics. Monoclonal antibodies in particular, due to their specificity, relatively long half-lives, and low toxicity, have entered the clinic to treat a variety of diseases ranging from cancer to inflammatory diseases. However, analytical characterization continues to confound many researchers. This webinar will offer some solutions. Panelists will discuss advances in instrument capabilities and techniques that provide reliable, consistent, and faster analytical methods. Several case studies to introduce the application of various analytical techniques for therapeutic protein characterization will also be presented. These techniques support comparability, similarity, and stability studies required during biological drug development. The two speakers today, Yi Du from Merck Research Labs and Ning Tang from Agilent, will share their experiences and insights in just a minute. Before we get started, though, I want to encourage everyone to submit questions for our panelists. We'll answer as many questions as we can after the final presentation. You would simply type your question into the Ask a Question box and hit Submit. Yi Du is a Senior Research Biochemist, Extended Characterization Bioprocess Development at Merck Research Labs. Yi will talk about characterization of recombinant monoclonal antibodies. He'll discuss the use of LCMS for mAb characterization and the challenges he has faced and attempted to conquer in his work, including heterogeneity of biological products and high throughput and automation. Yi, we're ready when you are.
All right. Thank you very much, Tamlin. Hello, everybody. So shall I move on to my slide? Again, thank you, everybody, for attending this webinar. I'm Yi Du. Today I'm very glad to present the work on the characterization of recombinant monoclonal antibodies for biological drug development. So here's a brief overview. So I'm going to give an introduction about the biological drug development, and then I will talk a little bit about the characterization testing versus release testing, and followed by 3 case studies regarding some specific examples on the monoclonal antibody characterization. In the end, I will give a brief summary. It has been predicted that in 2014 that the top 10 drugs, 7 out of the top 10 drugs are going to come from the biologic drug. Therefore, the biologics or biologic drug has become a very exciting area and a growth area for the in the drug field, in the pharmaceutical industry. And from this slide, it can be seen much clearly that from now on toward the year of 2016, most of the pharmaceutical sale growth are coming from the biologics, either from monoclonal antibody, therapeutic proteins, or vaccines. So it is becoming quite exciting for the biologics and biopharmaceutical industry. However, the biologic drug development, discovery, and development is not easy. It always normally takes about 12 to 13 years to come with one drug put on the market, from the basic discovery towards preclinical development to clinical development and commercialization. Since I'm from the biologic drug development area, therefore my talk is going to be focusing on the biologic drug development. It composed of several key functional areas as shown in this slide. It comes from the cell line development and the cell culture development and purification development, and also formulation development. All in all, the analytical development will support all these critical areas and to provide support for all this biologic drug development. Why we want to say that the biologic drug is however, biologic drug is very challenging project compared with a small molecule. Why we want to say that? Because it is a very heterogeneous molecule compared with the small molecules. Here are some example of where the heterogeneity come from. Mainly it's from this structure. This is a typical structure of IgG1 monoclonal antibody. It is composed of this heavy chain and light chain linked together by the disulfide bond. And the Fab region is mainly for the antigen binding, and FC portion is mainly for the to react with binding with FC gamma R1 receptor, and mainly for the ADCC or CDC function of the monoclonal antibody. Several examples of heterogeneity are listed on the left side. You can see that basically from the primary structure, there is an N-terminal pyroglutamate could happen during the storage, and asparagine and aspartic acid can easily be degraded, followed by deamidation or isomerization. And methionine on the molecule can be oxidized easily after a long time of storage. Other than that, during the cell process, the heavy chain C-terminal can be degraded and become some C-terminal variants can happen. So also, since there is glycosylation on the CH2 domain, therefore there are glycosylation variants that could happen. It's not only composed of a high mannose form, but also composed of complex form. Therefore, all this information has to be analyzed or characterized before for us to get a good understanding of the molecule before we can put it on the market. So next slide, I want to highlight, since a lot of these analytical activities are composed, can be mainly composed of the characterization testing and the release testing. So there are some differences between these two testings. So characterization testing is mainly required when we're establishing a gold reference standard of an internal product to get a detailed information of this molecule. And also it is required for comparability and similarity studies. However, it is often technical challenging, therefore it is hard to be applied in the QC lab for routine use. However, release testing, which is normally come from the characterization method, is after further refinement. It's basically needed minimum number of tests is required for the evaluation of this drug substance and the drug product purity, identity, safety, and potency. It is always it's developed and validated for routine use in the QC lab. So here are some examples of what we do for the characterization testing. To understand the primary structure, we have to do intact and reduced mass analysis. We have to do peptide mapping by the LCMS. And also we have to understand the glycosylation, the disulfide bond linkages, and N-terminal, C-terminal, terminal verification. For the secondary and tertiary structure, some biophysical assays have to be applied, like FTIR, far near UVCD, or fluorescence, etc. We also have to use some functional assays to understand the antigen binding, epitope mapping, and Fc gamma N binding, for example. Some other assays include reduced and non-reduced CESDS, CEX, size exclusion, PSC, and AUC. So these all you can see that is a very comprehensive analytical method can be applied to understand this monoclonal antibody as complete as possible, so that we can have a good understanding of the molecule. However, for the release testings, we can see that here are some basic requirements we would normally use. However, it also depends on the project. It's probably not all the release testing should be used for each different project. But normally for the purity, we have to look at the pure appearance, to measure the protein concentration and some visible particles, for example. And to get identity testing, sometimes we have to do a peptide mapping, or CIEF or CEX HPLC. For safety and potency, we all have different release testings. Today, I'm going to focus on the characterization testings. So you may want to know why we want to do such characterization testing. So here is one of the applications of how we use the results obtained from this characterization testings. So it can be used for assessment of CQAs for quality by design. So quality by design has recently been promoted by FDA to get full understanding of the relationship between the quality attributes of the product and their impact on the safety and efficacy. Therefore, it requires to get a good knowledge of the relationship between the structure and biologic functions. In that case, the characterization testing can provide such a good understanding of the molecule characteristics. And also, critical quality attribute is physical, chemical, or biological, or microbiological properties or characteristics that can be defined so that we can use them to contribute to the quality by design. Our characterization results play a critical role to evaluate these CQAs to support the quality by design. So I'm going to present some examples of how we normally do some characterization work in our lab. So here are two monoclonal antibodies manufactured from two different sites and obviously from two different processes. But they are the same molecule, have the same primary sequence. They both are IgG1 class. However, some observations were made that there is a significant difference in the CEX profile between the MAP-A and the MAP-B. We have to carry out some intact mass analysis and peptide mapping by LCMS to characterize the differences. So this is CEX of MAP-A and MAP-B. As we can see that it obviously has more basic species on the MAP-B compared with MAP-A. So we would easily think of that is because of the C-terminal lysine environment that contribute to these differences. And we applied the CPB, the carboxy peptidase B, to remove the C-terminal lysine, and the CEX profile looks similar after the CPB treatment. And to further confirm it is the C-terminal lysine, we carried out the intact mass analysis, and this is the instrument setup for the intact mass analysis. We can see the LC is Agilent 1290 Infinity UPLC, and mass spec we used is 6538, and the column and the temperature and flow rate were the basic experiment setup. So this is deconvolution result of these two molecules. We can see that MAP-A composed mainly without C-terminal lysine. Therefore, we can see the major distribution of the main Glyco forms. However, MAP-B, we can see obviously is become more complex. It largely because of the C-terminal lysine, and the mass matched very well with the C-terminal lysine, with either one C-terminal lysine on one heavy chain or two C-terminal lysine on two heavy chain. We also carried out the peptide mapping analysis after the lysine digestion. And this is, again, the setup we use. And this is the overlay of the UV trace between the MAP-A and the MAP-B from two molecule drug product. If we look closely, and from this UV trace, we can see that from MAP-B, there is a one peak showed up that matched very well with the C-terminal peptide with a lysine on the C-terminal, and it has very good mass accuracy. However, it didn't show up in the MAP-A profile. However, on the right-hand side, we can see that both MAP-A and MAP-B has this peptide without the C-terminal lysine. The mass also match very well. However, in the MAP-A, the signal intensity is a lot higher than in the MAP-B. this is a very good evidence to demonstrate that the difference it is from the system-less environment, and give us a confidence that there are some differences in these two processes, and we have to find out the reasons back in the process to see what we can do to make them look much similar. So here's another example. We want to characterize this MAP-A during their stability studies. So MAP-A is again another IgG1 monoclonal antibody. We carried out some research stability study in different conditions. Basically, it's a minus 80 degree and a 30 degree. We just want to give some accelerated stress condition under 30 degree. And over 6 months, we pick up the time point at 1 month, 2 months, and 6 months. We want to use this study to probe the degradation pathway of this monoclonal antibody. We did observe the deamidation on one asparagine increased at elevated temperature over time. So deamidation and isomerization, as shown in this slide, mainly happen on the asparagine and aspartic acid on the monoclonal antibody. It's a commonly observed degradation for this monoclonal antibody over the time, either during the storage or during the sample preparation, but sometimes. So as we can see that asparagine deamidation and aspartic isomerization all go through a similar pathway, that they form a succinimide intermediate, then followed by the water hydrolysis. They can form either isoaspartic acid or aspartic acid. However, the molecular weight difference between the isoaspartic acid or the aspartic acid and asparagine is only 1 Dalton. In that case, a high-resolution mass spectrometer is needed to characterize or determine this deamidation happening. So this is an example to show that asparagine deamidation happened by the LCMS after the trypsin digestion. So this is asparagine-containing peptide. As we can see that on the top panel, there is no deamidation. However, on the bottom panel, there is some deamidation happen, and isoaspartic acid always normally elute earlier than the main peak, and followed by aspartic acid after the deamidation. And their mass, as you can see, that isotropic distribution are very similar to each other. However, the isoaspartic acid and aspartic acid are 1 Dalton higher than the non-deamidated species. And this gives us a very confidence that it is happening on this sample. And after some relative quantitation, we can get a trend or change of this deamidation happening over the time at this elevated temperature. These two different bars are obtained to the data at each time point or condition comes from either UV trace or the MS-ESC. Therefore, you can see that at the T0, there's only within 5% deamidation happening. However, after 6 months at 30 degree, almost 20% of the molecule have deamidated. And there is no significant difference between the UV trace and the ESC data. So that tells us that sometimes we can use ESC to get the result if we don't have a good separation from the UV trace. Next example, I want to highlight some new technology development and its application in our process development. We want to, basically it is, we use this Agilent MAP Glyco chip to enable us to do some high throughput Glycan profiling. The reason we want to do this is that during our process development, there are various conditions need to be optimized. Therefore, high throughput or automation is largely required for us to get a good, quick, and solid decision making for the process development. However, for MapGlyco chip, we also care about its reproducibility and its result compared with our conventional 2AB normal phase HPLC assay. This MapGlyco chip is coming from, it has been recently been introduced by Agilent. It has to be used on this chip cube. It used as a LCMS interface to put in front of this QTOF. A chip, as you can see here, is largely been modified to have this PNGSF embedded in this chip and to allow a protein, a monoclonal antibody, to be sitting there and get to remove all the glycans and release all the glycans, and then all the glycans can be separated on the same chip and analyzed by the QTOF. I'm not going to talk in detail about how this MapGlyco chip works, because I believe Dr. Tang is going to give us a full description of this MapGlyco chip. But I want to highlight several points about how I use this MapGlyco chip. Basically, I just dilute this MapGlyco, dilute all the monoclonal antibody samples to 1 mg per ml with water. You can see that it largely minimized the sample preparation compared with 2AB labeling method. And then I can just inject 2 microliter to the MapGlyco chip. And the total runtime is around 12 minutes, and the data analysis time is around 2 minutes as well. And in that case, total analysis time for us to get a glycan profile is around 15 minutes per sample. I want to show you some data I obtained from this MapGlyco chip for one of our monoclonal antibodies. As you can see, after 15 minutes, this is a profile we got and also a table. I think one of the beauty part is that it also can do the data analysis automatically through this mass hunter, so that you pretty much just put the sample in and run the experiment and get the data like this. They give you a table to give you all the major glycan forms and their percentage in the total volume, percentage within all the glycans they have detected. Not only that, but also some of all the glycan forms can be identified very quickly by doing this database search, and this glycan can be identified fairly easily and visually displayed on the result report. So not like a 2AB labeling method, we have to run a standard and to get ID of these glycans. And all of the glycans we can identify it very easily. Also, you can customize your database to get a personal compound database library, so you can put any glycan forms you want in the database, so that you can search against this database for your own system, from your own species or your own system, so that you can get all the protein, all the glycans identified much easier. Later on, we also evaluate the reproducibility of this Glyco chip. This is run-to-run reproducibility. We can see that from 3 separate runs on the same sample, the variance is very small, and again, we see the same result from the day-to-day variation, that the reproducibility is fairly good. So this is, we compared our result with 2AB labeling method. However, we do observe some differences between this 2AB labeling and the chip analysis result. However, there may be some reasons. One of the reasons is that the sample size we used is fairly limited for either the 2AB labeling or the chip analysis. We only analyzed one sample. This is from one sample and one time. So the variation can be fairly large, and Also, the detection mechanism is different. So TWABE labeling is basically the fluorescence detection. However, the chip analysis is largely depend on the ionization efficiency of different glycan forms. So there might be some differences in there. Due to this reason, we might not be able to use this method, Glyco chip method, for any of the release testings, but we can still apply it to support our process development. Here is an example that we do use the Glyco chip to support our in-process development. So these are 5 different conditions in the in-process development to get these 5 different monoclonal antibodies, and their glycan form, the glycan distributions are so different that you can tell very easily. And I can get this result in about an hour or 2. That is amazing compared with a conventional TWABE labeling method. So because we can see that some of the conditions has all composed mostly of the high mannose form. However, all the other monoclonal antibodies composed of the complex form. The main reason is that some enzymes are prohibited. PNGSF actually is inhibited by some component we put in, some chemicals we put in for the process development. And therefore, the high mannose form are saved toward the end. And however, the other conditions, there are no such chemicals, and the high complex condition of glycan forms are formed. So this can give us a quick and good understanding of the process development. While some chemicals can make a significant impact on the glycan, on the quality of this monoclonal antibody, especially their glycan forms, and help us to optimize some of the conditions for us. Okay, so I'm going to give a brief summary. So basically, this extended characterization is required to support the comparability and the stability studies to understand the molecule and provide critical input for the CQA assessment, as I discussed above. And as we can see that this LCMS is a powerful tool for monoclonal antibody characterization in biologic drug development. Especially the high-resolution mass spectrometer is needed for more and more challenging characterization need. And also this new technique, Agilent Map Glyco chip, really provide opportunity for high-throughput glycan profiling to support this process development. Okay, last but not least, I would like to thank all my colleagues and in the extended characterization group to give me all the support to finish all this work. And also I'd like to thank my management on their sponsorship and their support my work to introduce this new technique in our group. And also I'd like to thank people from Agilent to give me all the support when I develop some of the method. And in the end, I would like to thank everybody for your attention. Thanks.
Thank you, Yi. Before we move on to our next speaker, I want to remind you that you can submit questions at any time by typing a question into the Ask a Question box on the lower left of your screen and hitting Submit. Our second speaker is Ning Kang, Senior Scientist at Agilent. She'll talk about strategies to accelerate biologics characterization, including the use of iFunnel technology. Ning, you can begin now.
Thank you, Tamlin. And for today's presentation, I'd like to talk about how to accelerate biologics characterization using the newest model, 6550 iFunnel QTOF. So this is the agenda of my presentation for today. I will first give a brief introduction to Agilent's toolbox for biopharma analytical analysis, and then with a focus of some detailed introduction to 6550 iFunnel QTOF mass spectrometer. And then I'd like to show some examples of intact monoclonal antibody analyzed on the iFunnel QTOF and some of the quantitation capabilities that can be achieved on this instrument. And then the second case study, I will be showing some oxidation analysis on the therapeutic protein and how users use the LCMS approach to help to solve the problems there. And then finally, I'll talk more about the MAP Glyco chip design and then its applications. So on this slide basically shows a lot of different analytical tools that Agilent offers that can be used in biotherapeutic development. So Agilent as a measurement company actually offers a very wide range of analytical techniques. So in this slide, I'm just showing a few of the examples. So for sample preparation, we have off-gel fractioner, which is showing on the upper left corner. And this is a device that allows user to separate proteins based on their PI. And then it's using an IPG strip to guide the separation based on PI, and the proteins actually will be in solution on top of the IPG strip. So this provides a very nice way for people to want to fractionate their proteins based on their charge variants, and then followed by further characterization of each different species. And then we also have here showing Bravo robot, and this can be used to do a lot of liquid handling, including automated tryptic digestion. And then more recently, we have also introduced an assay map suite of tools, which is highlighted here. It's a special type of liquid handling head that has functionalized tip attached to the head. So for these functionalized tip, we have protein A, streptavidin that people can use to do purifications of their proteins. And then we also have immobilized trypsin and also C8. So people can use them to do tryptic digestion and followed by cleanup. So this kind of tool allows user to automate a lot of their sample preparation procedures. Showing here on the right, we also have the newest newest member joined the Agilent family, which is the NMR, and people use them to do a lot of the structure analysis of their drug candidates. And then as for detectors, we also have some other options here. We have a bioanalyzer, which basically is a microfluidic device, allow people to analyze the size and the purity of their DNA, RNA, and proteins. So basically a very nice replacement for SDS page, and the results are digital, so you can easily be put into reports and into your electronic notebook. And then we also offers a capillary electrophoresis, which has its own position in biopharma industry, and some people use it routinely for Glycan analysis, so that's a very nice equipment there. And finally, comes to our HPLC and mass spectrometer families. So we have different models in this family. Today, I'll be focusing on the newest release in our QTOF family. So this slide shows zooming into the QTOF mass spectrometer. So for the 6550 iFunnel QTOF, we have listed a few of the specs here, and it can reach mass resolution of greater than 40,000, and then it can also has fast scan capability, allows user to do 50 spectra per second for MS scan, 33 spectra per second for MSMS scan. And then it also has excellent dynamic range, can reach up to 5 orders of linear dynamic range, mass accuracy less than 1 ppm for single MS and less than 2 ppm for MSMS. The biggest improvement for this newest addition to the family comparing to older model of QTOF is actually its improvement in sensitivity. So I'll go through some of the details. What are the specific features that has been implemented on this QTOF that made it so special. So first of all, front end, we introduced a dual Agilent jet stream source that actually have two sprayers, and one of the sprayer is used for reference mass introduction, so that allows the mass calibration to be automatically adjusted in real time, so that guarantees us a robust and stable mass calibration. And the next feature here is actually the iFunnel technology that has been implemented in the QTOF. So iFunnel technology has been introduced on our triple quad for more than 2 years and has received a lot of success in terms of great improvement in sensitivity. So I have the next slide to talk a little bit detail about iFunnel specifically. And this collision cell, after ions has been introduced into mass spec, has axial focusing capability that allows us to do very fast MSMS without crosstalk. And after the collision cell, we also implemented a beam shaper, so that has ion beam compression and shaping capability that guarantee the best resolution and sensitivity from the mass spec. And the back end, we have a 4 gigahertz digitizer and the analog digital converter detector that gave us guarantee for better resolution, better mass accuracy, also the biggest dynamic range. And then we also put in a new embedded processor that allows to do a faster scan in single MS up to 50 spectra per second. Of course, to achieve the highest mass resolution, we also have the longer flat tube that allows the ion to have a longer flat path, and then it gives us a better resolution overall. So this slide actually shows detailed spectra for the detailed graphics for the iFunnel in the front end of the mass spec. So basically, we made several improvements here. At the front end, we changed the capillary from a single bore to a hexabore, and then so that allow us to sample more ions from the very beginning. And then, of course, it also bring more gas and neutral molecules. And then during this ion funnel, we actually have 2 stages here, and they have differential pumping. So the neutral molecules and the gas molecules are pumped away, and the ions are focused within the ion funnel, and then the 2 ion funnels are off axis, so the ions can be guided through the ion funnel while the neutrals will be filtered out. So the biggest improvement we get from the iFunnel QTOF is the improvement in sensitivity. So here I show one example. This is the tryptic digest of a single protein, and then we're just showing the extracted ion chromatogram of one of the peptide, and the blue trace shows the signal we're getting from the iFunnel QTOF. The red trace shows the signal we're getting from a non-ion funnel QTOF. So you can see the increase in signal from the iFunnel QTOF has been almost tenfold comparing to the older version of QTOF. And on the right-hand side is the targeted MSMS mode that shows that we have similar signal increasement when we're doing MSMS as well. So next, I'll show some data on the iFunnel QTOF to measure the molecular weight of intact protein. 2 examples I'll be showing are monoclonal antibody and antibody drug conjugates. So this slide is basically the raw data of a monoclonal antibody measured in the 6550 QTOF, and this is the charge distribution envelope. And then if we zoom into one of the charge state, we will see that these typical finger patterns showing the glycoforms attached to the monoclonal antibody. And then this is the raw data. After deconvolution, we'll obtain the neutral mass, zero charge spectra for the monoclonal antibody. That's range from around 150 kilodalton. So here shows 4 different fingers for the glycoforms, and then you can see the mass difference between 2 fingers are around 162 dalton, which is typical glucose unit. And then this particular deconvoluted spectrum is on top of maximum entropy deconvolution. We applied peak modeling to the raw data and then use that to further validate the results and then get higher confidence of the deconvoluted proteins from the maximum entropy deconvolution. So you see a cleaner and higher confidence protein patterns measured from the 6550 QTOF. So other than qualitative analysis, we're also trying to assess the quantitation capability using our 6550 QTOF. So in this experiment, we did a serial dilution of the monoclonal antibody from 5 microgram down to 5 nanogram loaded on the column. So on the left, showing the raw data for each different sample, you can see the bottom one is the charge envelope we obtained from the 5 nanogram antibody loaded on the column. So there's still a reasonable charge envelope that we can recognize belong to an intact protein. And then after deconvolution, the result is showing on the right, and we can see the glycoforms from the antibody, and that, of course, once we go to lower concentration, start to the qualities become poor, and that's expected. So to evaluate the quantitation capability, we use the peak area from the deconvoluted results and then use them to try to have a linear fit. So the result is actually showing on this slide. This slide basically shows for each different individual glycoforms, we're quantifying from 5 nanogram up to 5 microgram on the column. And the plot is a log-to-log peak area versus the concentration of the proteins. So you can see clearly down to lower concentration. So we have shown very reasonable linearity from 5 nanogram all the way to 5 microgram, demonstrating 3 orders of magnitude dynamic range for intact protein quantitation on the QTOF. So this could be very helpful for people who want to do some relative quantitations at intact protein level for their samples. So the next example I'm showing is antibody-drug conjugate. Antibody-drug conjugate can be way more complex than a native antibody. So this is the charge envelope. You can see the sensitivity has dropped because we have a more complex sample. And if we zoom into some of the charge states, you can see that the resolution become poor, and then you have actually more peaks within each charge state, and that indicating different number of drugs attached to each monoclonal antibody. So overall, the protein sample is more complex. So after deconvolution, we get such a picture, and that shows different clusters of proteins measured from this sample. So if we look at them, within each cluster, the mass difference between 2 peaks are about 162 Dalton, which indicate the glycoforms. And then we have 4 different clusters, and then the mass difference between each cluster is about 1,481 Dalton. So that indicates the drug actually attached to the native monoclonal antibody. So from this sample, we can detect the naked monoclonal antibody and the map with 1 drug linked, with 2 drugs and with 3 drugs. So in such a complex sample, depend on the extent of the chemistry, and sometimes you can have some fairly low abundant species representing different number of drugs attached to it. So it's important to have better sensitivity for the mass spectrometer in order to be able to have a correct picture of the drug to antibody distribution in this for this particular sample. So the next case study I'll be talking about is oxidation analysis for therapeutic protein. So in this particular study, the protein we're studying is antibacterial therapeutic protein P128. Recently, the bacteriophage-derived therapeutic proteins has become an important class antibiotics due to the development of resistance to antibiotics by certain strain of bacteria. So P128 is bacteriophage derived. It's Staphylococcus peptidoglycan hydrolase. However, the active site is unknown at this point. And this protein is expressed in E. coli, and the molecular weight is about 26.4 kilodalton, as shown in the first spectra here. And then in this particular oxidation study, the P128 is subject to different stage of oxidative stress by applying with different concentrations of oxidizing reagent. So here shows two of the conditions. When P128 is oxidized with 10 to the third ppm oxidizing reagent, we can see that mostly it still maintains the naked form. However, we start to see small populations carrying 1 oxidation, 2 oxidations, 3 oxidations, and 4 oxidations. When we stress the protein with high concentration of oxidizing reagent and measure the molecular weight, we observe that the naked protein almost disappeared, and the main species is actually the protein carry 3 oxidation sites on it, oxidation modification on it. So to further evaluate the drug activity versus the oxidative stress, they have conducted an experiment. In this experiment, the bacteria is grown up to a certain concentration, and then 10 to the 7 cells are aliquoted, and then different P128 therapeutic protein subject to different degrees of oxidative stress will be incubated with the cell. And then after incubation, the residual cell number will be counted. That represents the effective of the drug activity. So this graph, the y-axis is the residual cell count, and the x-axis shows different conditions that's been treated with the cell. So the first bar actually represents the cell control, which means the cell is not incubated with the drug. You can see that we have about 10 to the 7 cells. And then when it's treated with P128, you can see that the cell number greatly reduced, so which means the drug is active and then inhibiting the growth of the bacteria. And similarly, when the P128 is subject to mild oxidative stress, that doesn't affect the drug activity, so it's still active, controlling the cell growth. When P128 has been extremely stressed by oxidative conditions, we can still start to see the drug start to lose activity, and then until it almost lose all the activities. So this is the relationship between oxidation stress versus the drug activity. And then to further analyze which amino acids actually contribute to the deactivation of the P128, the researcher has done tryptic digestion of different proteins and then did peptide mapping using the LCMS device. So in this experiment, we have 6 different conditions, P128 with under different oxidative stress conditions. And then after tryptic digestion, each sample we have 5 technical replicates that's indicated by the same colored dots. And this plot is a PCA plot that's by importing all the peptide mapping results into such a statistical analysis program. And then what it does, it actually shows grouping of the relationship between different experiments, and that helps to tell us which samples are similar and which samples are different from each other. So this is a very fast way to analyze large batch of LCMS data. So in this, to further look into the details, first we notice that the same color dots actually cluster together. So this is a good sign demonstrating good reproducibility of our technical replicates. And then the samples that has been severely stressed by oxidation conditions are located down to the bottom of the plot and separate from the other conditions, indicating they are similar to each other. And then we have also demonstrated by the bioassay that they have lost activity of their particular activity. So in this from this PCA plot, we then can identify which particular LCMS runs we want to look further. So in this case, we have picked a control sample, which is the native P128, comparing that with one of the sample that has 10 to the 7 ppm oxidizing reagent treated. So in this particular program, then we can compare sample 1-to-1. So we first get a tabular view, which listed all the peptides identified from both experiments. And then the two experiments are color-coded. So the control reference is pink, and the sample is blue. So it listed all the features, properties has been identified from these two LCMS runs. So when we click on each individual peptide, then we also have a chromatogram comparison down to the left bottom corner, and also the MS spectra and MSMS spectra associated with each of the peptide that has been identified. So by just a single click through the table, that allows us to quickly go through two samples and look at the differences between these two related samples. So here shows an example of a peptide that without oxidation on the methionine, showing in the middle spectrum, and then also the same peptide sequence with methionine oxidized, showing at the bottom of the slide. So comparing them, this is the MSMS analysis. We can identify a series of B and Y ions. Also, after Y5, we start to see a shift in mass of 16 Dalton, which indicates the oxidation at that particular amino acid. So to summarize the entire experiment, we have identified two tryptophans that are actually critical to the activity of P125. These two tryptophans are close in the sequence. So we have identified from this structure the location of this stretch of sequence, and that has been hypothesized as the active site for substrate binding for this particular therapeutic protein. And then mutagenesis study is being carried out right now to further validate this hypothesis. So the last case study I'll be showing is to talk about MAP glycochip. So Dr. Du has mentioned about the MAP glycochip, so now I'd just like to give you more detail about the design of the chip. So this shows the picture of HPLC chip, and then it's the size, the real size is about the size of a credit card. So outside is a metal frame, inside the dark part is actually an inert polyimide film, and then we use laser to ablate channels, microchannels on the films, and then different layers of the polyimide film will be laminated together to form the microfluidic channels. And then in these channels, we then can pack, for example, C18 material for peptide mapping, and then C8 for protein analysis. And then not only we have analytical nano column, we also implemented enrichment column on the chip. In the middle here, the dark circle shows the enrichment column. And then at the front end of the chip, we also pull out a nano spray tip. So this really has zero depth volume post column, which is quite important for nano spray analysis. So with the HPLC chip, it's widely used in proteomics field, and then people can use that to do intact protein analysis, peptide mapping, PTM, and now with the new MAP glycochip, people are able to do Glycan analysis for monoclonal antibodies. So traditionally, for Glycan profiling, people use LC or CE separation with fluorescence detection. And this workflow usually takes 2 to 4 days. And then recently, people have been using mass spec as a detection method for Glycan profiling. Multi-MS has been used. And then in this particular workflow, it's usually 3 to 8 hours. And then the MAP glycochip we're introducing here actually can complete this analysis within 10 to 30 minutes. So the way we implement the chip is we have 3 columns integrated on a single HPLC chip. And the first column is immobilized PNGSF, so that we call an enzyme reactor. So because of the high concentration, local concentration of the enzyme, and that really favors the reaction. So we typically can achieve a fully deglycosylation within a few minutes. And then after the glycans has been cleaved, they will be enriched on the graphitized carbon glycan enrichment column, and then followed by separation by the graphitized carbon nano column, and then sprayed right into the mass spectrometer for detection. So here just one experiment showing the speed of deglycosylation using the PNGSF enzyme reactor. So here shows a monoclonal antibody before deglycosylation. We see the typical glycoforms, and then after 3 seconds deglycosylation on the chip, we can see that removal of one set of the glycans, and then also some of the removal of both set of glycans from both heavy chains. And then after 6 seconds deglycosylation, we see the majority of the monoclonal antibody has been fully deglycosylated. That results in a single peak matching the mass of a deglycosylated monoclonal antibody. So this slide shows the workflow for the MAP Glyco chip. So basically, the MAP Glyco chip has two big steps. The first big step is sample prep. That includes sample loading, filling of the enzyme reactor, and the incubation time for deglycosylation. That's typically set as 6 minutes in total. And then after the deglycosylation has completed, the glycan is transferred on the enrichment column, and then followed by a separation column. That can be completed within 6 minutes as well. So this spectrum shows the typical results from the MAP Glyco chip, and then we can see that the first 8 minutes are empty because we're mostly doing sample preparation and transferring the glycans onto the analytical column. And then all the glycans are eluting between 8 to 10 minutes. And then if we zoom into this retention time window in the insert here, we can see probably a dozen of different glycans have been identified. Although they're not baseline resolved, but because of the mass spec detection, we're still able to identify them. So once we acquire the data, we have an automated data analysis method, and that can automatically search against database and then identify all the glycans, and that can be put out into a report with the relative abundance of each glycan species. So Dr. Du has mentioned that we have a personal compound database and library package. So shipped with this MAP Glyco chip, there is a monoclonal antibody glycan database that already has more than 100 entries for the chemical formula and accurate mass for the glycans. And then on top of this, the user can further adding their own glycans they have observed that's unique to their sample. And then once the customer has fixed the retention time, they can start to record the retention time into this library, and that allows the user to search not only according to accurate mass, but also according to retention time. And then further, the people can take advantage of a Q-top mass spectrometer to perform MSMS analysis on the glycans, just to further validate the identity of their glycans. So in this library, you can attach different MSMS spectra acquired on different glycans. And we have showing here is one example of a glycan MSMS spectra acquired and attached in this library. And then in the future, you can perform MSMS experiments on an unknown sample and then use that to match to existing known glycan database for MSMS validation. So here I have more data to show the reproducibility of the MAP Glyco chip. So this experiment was done on 10 separate chips, and then for each chip we have done 250 consecutive injection of a monoclonal antibody, and just to see the reproducibility and the lifetime of the chip. So in this, on the left, showing the glycan profiles from the first injection comparing to the number 200 injection, we can see very similar glycan profiles and relative abundance, which shows the chip has been holding well in this 250 injection runs. On the right, we also plot individual glycans. Some of them are high abundance, some of them are relatively low abundance. Their relative intensities throughout 250 injections. So we can see that they basically all have less than 5% RSD for all different glycans. So here is a comparison study we did using MAP glycochip compared to the traditional CE fluorescence detection and also the multi-MS experiments. So in this particular experiment, the same monoclonal antibody was used, and then for multi-MS instrument, there were 34 replicates. For CE lift, we did 43 replicates, and then we also ran 3 separate sequences on the MAP glycochip. Each sequence contained 50 replicate runs. So each experiment is color-coded, and then we can see that for some of the major glycans, we have fairly consistent results for relative abundance obtained from 3 different technologies. Also, the CV for 3 different techniques are very tight and very similar. Of course, when you go to some of the lower abundant glycans such as G0F or MAN5, you start to see bigger variations among different techniques. That's typically observed when you reach the getting close to the detection limit of each analytical technique. You start have more variations in the value you observe. Okay, so I'd like to summarize my talk today. I have shown you that Agilent offers a broad range of analytical tools for biopharma analysis, and I've specifically shown you the 6550 iFunnel Q12 has excellent sensitivity and quantitation capability for intact protein analysis, and also the MAP glycochip has been proven to be a robust technique for high throughput analysis for MAP glycan analysis. So with that, I'd like to acknowledge the people who have contributed to the results I've been talking today, and people from Genentech has given us the initial idea and then participate in the development in the MAP glycochip. Also, we have very wonderful collaboration with Gangagen Biotechnologies on the oxidative analysis. And we have a group of people from Agilent also contributed to different projects that I've been talking today. So with that, I'd like to thank you for your attention.
Thanks, Ning. Before we begin the Q&A session, I want to encourage everyone one final time to submit questions for our panelists. Please type your question into the Ask a Question box and hit Enter. Now we're going to start our Q&A discussion. We have a lot of good questions already, and we'll answer as many as we can. Before we start the Q&A, I want to let you know that Yi-Du is unable to join us for the questions due to a traveling commitment. Any questions directed at Yi will be relayed to him, and he'll answer as many as he can. Ning is with us, and she will answer as many questions as possible. And Ning, I hope you're ready because we do have a lot of questions. The first couple all relate to MAB glycochips. So here we go. Is it possible to use MAB glycochip on other glycoproteins other than MAB?
Okay, so the MAP glycochip, we recommend it to be used with monoclonal antibody. Part of the reason is that monoclonal antibodies' glycosylation sites are well exposed, and the deglycosylation process is better controlled. We also have tested dozens of monoclonal antibody, and they all reach full deglycosylation within a couple of minutes. So we have this workflow nailed down pretty well. Different glycoproteins may need different denaturation steps before deglycosylation steps, and individual glycoproteins may present very different kinetics in terms of deglycosylation. So having said that, there are users using the chip to study various glycoproteins other than monoclonal antibody, as well as some compact samples such as serum. They just want to take advantage of the speed of MAP glycochip. Of course, optimization is highly recommended if you're using the MAP glycochip for other glycoproteins than the monoclonal antibody.
Okay, how long is the equilibration for the MAB glycochip?
Okay, so if I understand equilibration correctly, it's the incubation time for the monoclonal antibody to have contact with the PNGSF. That is set as 4 minutes as the current protocol.
All right, and then can the MAB glycochip be used with any LCMS?
MAB glycochip, it has to be used with the HPLC chip cube, which only works with Agilent LCMS.
Okay, here's another one. Is MAB glycochip interchangeable with 2AB labeling approach for Glycan analysis?
So we have done some benchmarking studies comparing the results with 2AB labeling versus the MAB glycochip. So we have relatively consistent results throughout these two methods. So we believe technically you should be able to interchange the 2AB labeling approach with the MAB glycochip approach. However, they do have different advantages and disadvantages. So you might have to do some validation when you're doing that.
Okay, and how pure would a protein sample have to be analyzed with the glycochip?
The typical usage is for monoclonal antibodies, so it's usually very pure. However, I did mention that we have a couple of users using it with different samples, including complex samples. The purpose of their experiment is not characterization of the glycan profile of any particular glycoprotein. They were doing biomarker-related studies. So in order to characterize a glycan profile from a glycoprotein, monoclonal antibody, it has to be fairly pure.
And now we're off the MAB glycochip and on to some other questions. What buffers did you use for quantitation of MABs from 5 nanograms to 5 micrograms? Okay, so in that particular study, I basically did zero dilution of the monoclonal antibody into 3% acetonitrile and 0.1% formic acid. So we also have some data that we didn't share today, is a similar quantitation experiment that was done by spiking in the monoclonal antibody into serum samples and then pull down using other proteins, and that was just in the buffer, in the pull down buffer. So we achieved equivalent quantitation capabilities from that experiment as well. Okay, is it LCMS or direct MS?
So all the data showed today are obtained from LCMS.
Okay, and then the next question is, have you ever seen the sialic acid containing glycan by this method?
Yes, we did. So we have seen sialic acid containing glycans from this approach. We do realize that when you have multiple sialic acid, then you're adding more negative charge. So this MAB glycochip current method is done on in positive mode. We also have another protocol which potentially can allow people to use the chip in negative mode, which may have better ionization efficiency for multiple sialated glycans.
Thank you, Ning. Alas, our time is up and we don't have any more time for questions. Thanks for listening in and thanks for the great questions. This webinar will be archived for 6 months on our website, www.genengnews.com. If you missed parts of it, you can watch it again or you can recommend it to friends and colleagues. Once again, I want to thank our panelists as well as our sponsor, Agilent Technologies. We'll be sending you a survey shortly and hope you'll take the time to respond. Your comments will help us continue to provide topical and timely webinars. Thank you.